So, now let us get into the preliminaries of this subject. So, first we look at the organization of it. At the lowest level of the body, that we get the cells are there. So, which are the basic constituents of the human body, and in a human body for an adult we have about 100 trillion cells, and it is more interesting to know that out of them that every day 50 million they dies, and another 50 million comes back, actually are generated to take their position, so that the body does not degrade or degenerate. Okay. So, that is the way they actually make up for the loss, and the cells are the basic building block. They are full of that the fluids, and we can also say that outside the cells there are fluids. So, you can say they are also floating in the liquid. So, half of the body is actually made of liquids, and cells of a particular type in terms of their that function as well as their constitution, they form the tissues. Okay. For example, we have that muscles, we have that skin. So, these are different kind of tissues are there. So, then multiple tissues, that means 2 or more tissues, they gives rise to one functional unit, that is called organ. For example, kidney, or we can take the pump in the body, that is heart. So, these are the organs, and multiple organs together they give rise to one subsystem. So, subsystems are like that what we covered in the previous talk, that we told about the digestive system, nervous system. So, there are a lot of subsystems like that, and all such subsystem when they act in a harmonious way, we get the existence called the human system. Okay. So, this is very important for us, and we would like to learn about it, and so we will go ahead with this thing. So, next we would like to look at the human cells. So, first let us look at in the scale that of that the size, that the smallest things could be the molecules which are inert, the small molecules they are inert. Then we get the virus little bigger than them, and they are in the border of actually the living being and the inert objects. Okay. Outside they cannot procreate. So, they are like inert objects, but once they get into a cell they can actually create the replica and create a very difficult situation for the cell to survive. Then we have the bacteria, they are living being, and after that the animal cell or the human cell for which we are interested, and little bigger than the human cells are the plant cells. Okay. And here usually when we talk about the cells, we use this unit Armstrong, which is 10 to the power minus 10 meter. So, in terms of Armstrong usually we talk about, and we get this cell is that animal cell is in diameter about 20 the micrometer, and it is actually covered by, it is surrounded by one semi permeable membrane, and within this membrane we have some liquid called cytoplasm, and outside also some liquids are there. Okay. That is called that interstitial liquid we can say. So, both the sides the liquids are there, and within this we have lot of objects, some of them they are living, they are called organelles all together. For example, mitochondria, that is the powerhouse of the body, and then another organelle is actually the nucleus, which is the controlling unit. Apart from that, there are many inanimate objects, like we have the proteins, we have the starch, which are required for actually generation of the power as well as for the building of the cell or repairing of the cell. And within the nucleus, that we get first of all a boundary, and inside that there is we get some fluid called nucleoplasm, and we get some circular objects within that. So, they are called nucleoli. They are filled with RNA or ribonucleic acid, and this nucleus also carries something very important, which are DNA. That DNAs are the keys, that whatever the characteristics of the cell is there, or for that person, that the characteristics are there, that is encoded in the DNA, and these DNAs are, or the genes are actually consisting of the DNA or deoxyribonucleic acid. So, this is in short about the cells. Now, we should move forward, we should go for that how the cell actually acts as a living being. So, for the living of the cell, metabolism is important, or in simple words we can say we need to release the energy what is required for the operation of the cell. And for that what we need say glucose for liberating the energy and the oxygen, and as we need them and we need to burn them in the powerhouse is mitochondria, which helps to actually release that energy. After that we need to, for that to continuously generate that, we need a supply of these two things, the glucose and that oxygen, and also after that the end product that is generated out of it, that is carbon dioxide, we need to get rid of the body. So, all these things we need to do. So, we need a transport mechanism for that, and this transportation of different things are primarily happening, or most of the part is happening through diffusion, and it is a passive process. The first thing we should note that it is a passive process, this our diffusion, and how it works. The first thing which makes it possible, that we told that cell membrane, it is permeable or semi permeable, we would say. So, that permeability allows for that diffusion of different kind of that ions and particles. That is the first point. Second point is that diffusion is possible because there is a difference in relative concentration. That means, inside the cell and outside the cell, for a particular kind of say ion, that must have difference in concentration. Without that, diffusion is not possible. In fact, in the nature there is a tendency to go towards the lowest energy state, or the what we call that equilibrium, and for that what happens, that from the high concentration some of that particles, or you can say that ions, they would leave and go towards the lower concentration. So, they are trying to go towards the uniformity or balance in concentration. And another thing comes into play in this diffusion, that is the electrical impulse or electrical potential, I would say. And how that happens when we are talking about the diffusion of the ions, and there is a imbalance of charge inside the body and outside, inside the cell body and outside the cell body, that gives rise to some potential difference. Now, that potential difference or that potential gradient it actually motivates some of the, actually repulses some of the ions and attracts some of the ions. So, they also influence the process of diffusion, and in the latter part today we will see that how that actually take part in the diffusion. Apart from the diffusion, we have that other two things, that active transportation, which is an active process, and we have a specialization of it called pinocytes. In these active processes, that the cell spends a little amount of energy, whereas for the diffusion no energy is spent by the cell. So, now we will look at, go to the next part. Let us see that how this diffusion takes place. So, here we have a hypothetical cell diagram. The left hand side, that we have the intracellular fluid or the cytoplasm we can say, and outside the fluid it is the actually the liquid of the, that we can say that that interstitial fluid, or that the fluids which is stored in the tissue. Now, both of them, they have concentration of sodium, potassium, chlorine and other anions. Inside the cell we get actually the concentration of the potassium ion, and the bigger anions, they are more compared to the outside, and outside the cell we have more concentration of sodium ion and chlorine ion, and because of that there is actually a concentration difference in two parts, that means within the cell and outside the cell. And cell membrane being semi permeable, it allows the movement of the potassium ion, say from the higher concentration to the lower concentration, that is meant outside, and for that sodium ion to go from outside to inside. However, that cell membrane is not actually impartial in this game. It has an important role, and what we find that it is very much partial. The way it is allowing the potassium ion to get out of the cell, they are not allowing the sodium ions to get inside in the same way. And as a result of it, what happens that inside the cell, that we lose that positive ions, and it gets into a state of actually negative potential. And what happens in that case, as it gets into the negative potential of the range of minus say 60 to minus 90 volt, or sometimes if you are asked to tell about one single voltage, then we can say minus 70 microvolt, millivolt. So, that kind of voltage that we get across the cell membrane, that negative potential now, it would actually resist the flow of the positive ions outside. So, a equilibrium is set, this electrical potential that is forcing the or repulsing the potassium ions to go out, and we get an actually imbalance like this, that the 10:1 concentration of potassium ions from inside to outside, and just the opposite for the sodium ion, that 10:1 from outside to inside, the concentration difference actually we get, and this potential is maintained so long this equilibrium is not disturbed. Okay. And that is why we call this potential as a resting potential. Okay. Now, let us see that how this actually equilibrium can be disturbed. Now, if there is a stimulus given to the cell membrane, which could be an electrical actually stimulus or a chemical stimulus, suddenly from one point that the permeability of the cell membrane changes and it spreads all over the place, and cell membrane suddenly becomes sensitive to the actually the plight of the sodium ion, and they start allowing them to come inside. And that happens naturally by diffusion, because outside the sodium ion concentration is much more than the inside. And at the same time, the potassium ions also, they are getting out of the cell, but it is not coming out in the same rate. Now, as if they are favoring the sodium ions more to pass the boundary. So, now because of that inflow of the sodium ion, the negative potential within the cell, that keeps on increasing and it stops to a actually level when it reaches about 20 millivolt or plus 20 millivolt with respect to the outside. And this process of change in voltage we called as depolarization. Okay. This is a depolarization. So, we call it depolarization. Next, once it reaches to that plus 20 millivolt inside, then suddenly that the characteristics of that cell membrane changes, and again it becomes impervious for that sodium ion. So, what happens at that stage? Now, it is allowing only the potassium ions to get out of that cell membrane. So, as they leave the cell body or the cell boundary, again the potential inside the cell starts dropping, and it can go as low as minus 90 millivolt. And this next process, it is called repolarization. And here in this, along with the diffusion, another thing we should actually keep in mind, that there is something called sodium potassium pump, which actually is an active process where it is spending some amount of energy to take 3 sodium ions inside and pushing some potassium ion outside. So, they are taking some sodium ion and throwing out the potassium ion. So, that is the way they interchanging the thing. So, that is called the sodium potassium pump, that is an active process. So, let us go next. So, here we show that the electrical process over the time, that the change in the potential actually is recorded over the time. We are looking at the change in the potential of the cell inside the cell with respect to that outside fluid. So, initially it is at the resting potential, and as some stimulus is added here, suddenly the characteristics or the permeability of the cell membrane changes and the depolarization starts, and it reaches to a level of about plus 20 millivolt inside, and then it stops or a equilibrium is reached, and after that it does not stay there for much more time, that the reverse journey starts and it goes through the process of repolarization and coming actually near again the resting potential. In fact, there is an overshoot which is called that hyperpolarization, this phase, and it slowly goes back to the resting potential. And here another thing we should know, that for the muscle cells the depolarization phase is associated with contraction, and this electrical pulse as it is going through the body, that is important for many activity, and the signals what actually the nerve carries is through this process of electrical activity. And how that send the signal, as the nerve signals along the length, this current actually flows from inside the body through the cytoplasm and outside the body through the interstitial fluid, and that is how the current flows or the signal flows from that one point to the other along the length of the nerve. And now we look at the characteristics of this potential. So, first let us look at that the nature of this potential. These cells now what we get, that it act as a small battery, and the source of this battery or the energy is the ionic voltage. That means, the current flow what we get, that is happening because of the movement of the ions or the charged particles, okay, just like any that battery what we use. Next, if we look at that the rate of flow or the transition of the action potential or the frequency of the action potential, that it is a very delayed compared to the electronic circuit. Okay. And here another thing we should tell, that the action potential, probably we should go back one step to tell that that here, that when we are talking about this action potential, once a stimulus is given, when it is going through, there is no way to stop it, and the amount of this voltage, that say minus 70 millivolt to plus 20 millivolt, this change it is not depending on the stimulus or the level of stimulus. When a stimulus is applied, it is like a threshold function. That means, the stimulus should be above some thresholds. If it is above that threshold, this depolarization will start. Now, once it is started, you cannot stop it till it goes to the end. However, by applying much higher actually stimulus, before it completes, again you may start it there. Okay. That is very rare phenomena. So, we can take as a simple explanation that it is a like a threshold unit. So, once the signal goes above the threshold, this action potential is created, and the voltage levels are independent of the input if the signal crosses that threshold, and you cannot stop that also in between. So, that is one of the important thing, and let us go back to that other characteristics of it. So, it gives rise to a voltage waveform, because it acts like a battery, and there is a coordinated electrochemical activity of large number of cells to get this biopotential or to record it. As we get that each of these cells, they have very little amount of energy or very little amount of current they can provide. So, if we try to measure that activity or record that activity, so we need some certain amount of actually power, and that is possible only through that coordinated activity of large number of cells. And there are a large number of such signals generated out of this active potential of the cells. They are say electrocardiogram, or what we know as ECG or EKG, then electroencephalogram, that is more known as EEG, electrogastogram, electromyogram. So, there are a number of them, with us. So, now let us move forward. Now, we see that what are the things it can do, that when it moves through the nerve fiber, the nerve fibers, they are actually protected by that myelin sheath, and after a small interval they are actually, for myelinated nerve there is some gap, and that when it is covered by the myelin sheath, the myelin being actually lipid or fat, they are actually acts as a electrical insulation and does not allow the transfer of the ion through the the cell membrane there. So, wherever the gaps are there, the exchange of the ions can happen there. So, the electric potential actually jumps from one such node to the other node, and that is how it actually moves through the across the length of the fiber. Okay. And for the myelinated nerve, such kind of conduction is called saltatory conduction. Okay. This particular name is given, which makes it possible that the fast conduction of the signal. So, we have just covered the action potential. Now, we complete that a small part here, and thank you for being with us.